They're on the outside of the double helix. We will highlight the backbones in blue. Now let's look at the crossbars connecting the backbone. Each crossbar is a hydrogen-bonded pair of bases, one from each DNA strand. An adenine is always paired with a thymine and a guanine with a cytosine. The double helix can also be represented by a ball and stick model. For simplicity, a ribbon model is often used, with ribbons for the sugar phosphate backbone.
Somebody guess the ribbon one after this? Question? All right, so here it is. Here is your sugar phosphate group here, your nitrogenous bases. There's your hydrogen bond, and they always, you'll notice, they always go with that pair. Here's the computer model. All right. So, if I have this as my one side of my DNA, what's going to be the other side? So T goes with what will be T. T goes with
A.
T goes with A. A goes with T. G goes with C. What? C. Okay. G goes with C. D goes with G. And T goes with A. And do you need to be able to do this? Yes, you do.
No, the test, if there's something like that, we have to match up. G, C, T.
Yep. Okay. Well, when proteins are heated, they usually denature. People start to come apart. If moderate heat were applied to this molecule of DNA, what pair or what part of the molecule would break down or break apart first? Use your knowledge of the chemical bonds. So look at your ABC. Don't put anything up yet. Ready, go. I have like one with like 3. The nitrogen base, nitrogenous, the nitrogen base pairs separate in the interior of the molecule. Why? Because those are those hydrogen bonds. Okay, RNA and DNA are polymer, just means a big molecule made up of little what? So my monomers, what are your little bits? Capital 10, so let's think it through. What are they made of? What are their building blocks? Don't show anything yet. Be careful. What is it? 2 nucleotides. Nucleotides make nucleic acid. They're very close, aren't they? Nucleotides build nucleic acid. All right, on that part, on my little double helix here, a small segment of that DNA molecule that specifies the production of a particular protein is called a gene. This is the stuff that builds you. You have a certain part on your DNA that is going to say, okay, this is their color of eyes. They're going to have freckles. This person isn't going to have freckles. This person's going to be tall. This person's going to be short. My family's short. I can blame it on my great-great-grandparents. My brothers wanted to be 6 foot. They didn't have a chance. Yeah.
My question is, on my mom's side, everybody's tall. On my dad's side, everybody's short. I'm confused. I end up in between.
That's usually it. Actually, I was trying to remember what's more dominant, tall and short. You know, it has to do with what dominates. Things like, and we do have our last unit is on genetics, which because it applies to us, everybody really enjoys that one. Things like skin color. It's actually a combination of 3 different genes. You can get 3 from mom and 3 from dad. So there's this whole palette of combinations from the very, very white to the very, very dark, and everything in between, and whether you get freckles and whether you sunburn and all of that's in there. How much melanin usually goes back to the roots. You know, your ancestors came from something that was a lot of sun, they protected themselves. Your grandparents came from, great-grandparents came from areas where they didn't have a lot of sun, like Norway, then you are here. Okay? It all works out, doesn't it? Anyway, it's fascinating to me. It's fascinating. And we'll talk about that. We'll talk about what happened. Okay. But you're right. Both my mom's family and my father's family are short, so we didn't have a chance. Okay. The activity of the protein is responsible for the trait associated with that gene. Okay. So when you're actually making that protein, again, what color hair you're going to have, all of that. And there's so many more things, whether you can digest lactose, you know, break down milk, or whether you can't. If it's in your family, how many do lactose intolerances in the family? And it's everywhere, isn't it? And once it's there, then you've got everything it's there. The key to DNA's function as an information molecule lies in the sequence of those nitrogenous bases, A, G, T, C. You change just a little bit, and you have a whole different thing. Just one little change, and the blood is just a little bit different. Okay? The DNA sequence, CCGATAC, determines the sequence of those amino acids when you are building the protein. Okay. A little like the blueprint. Is it going to be a 3-story house? Is it going to be a little 1-story cottage? Are we going to make a 50-foot tall skyscraper? It's all in the blueprint, isn't it? Yeah, let's start by DNA replication. DNA replication is to produce a new cell. So I have a cell, and up here in my nucleus I have my DNA, right? Well, when I want to make new cells, I'm going to make a complete copy of the DNA. I have to make a copy of every little page of the blueprint. Everything from eyelashes to kidneys, toenails, okay? And when you're done with DNA replication, you're going to have 2 full sets of DNA for each of your new cells. Now, do I want variety in these new cells, or do I want them to be exactly the same? So I'm growing hair. I'm growing hair. Do I want it to be going crazy, or do I want it always to be hair? I want it always to be hair. So DNA replication. You are making a copy of the DNA because new cells are being made. When there's change in them, then we don't want that. DNA is what we call semi-conservative. Remember, the body is always looking for ways to save energy. So DNA is what we call semi-conservative. Okay. This is where the new DNA molecule contains 1 parent strand and 1 new strand of DNA. So up here in my nucleus, there's my DNA. Now during DNA replication, it's going to open up or unwind the DNA, and it's going to come in. And remember, we said for every nitrogenous base it has a partner, and it's going to make partners, and it's going to build the new DNA. And what you have is the original parent DNA, and then you build this new, we call it daughter DNA, because this opens up and we're building, goes wrong, that actually should be bigger, right? We're now making copies. Instead of taking the energy to say, okay, I want my original partner, and you guys start breaking apart, because it will take energy, wouldn't it, to break these apart once they're built. Instead of taking the energy to break these apart and do daughter with daughter and parent with parent, it just stays. It has one parent strand and one new daughter strand, and it will go back and it will, reform the nucleus and go back in there. It doesn't waste the energy of doing that. All right, so let's see if we can make it more simple.
In principle, copying DNA, a process called DNA replication, is very simple. The two complementary DNA strands separate, and because each nucleotide can only pair with its complement, adenine with thymine and cytosine with guanine, each strand can be used as a template to build a new complementary strand, producing two DNA molecules.
In the cell, DNA replication is a little more complicated, but the principle is the same. For clarity, we have untwisted the double helix. Remember that each DNA strand has a 3' and 5' end, and the strands run in opposite directions. DNA replication begins at specific sites called origins of replication.
You don't need to remember this, just be
Proteins
aware of
attach
it.
here and separate the DNA strands, forming replication bubbles which grow in both directions. Enzymes called DNA polymerases move along the template DNA strand and catalyze the elongation of new strands. Because DNA polymerases can only assemble new DNA in the 5' to 3' direction, only half of the new DNA can be synthesized in one continuous piece. The other half is synthesized in short pieces. As the replication bubbles grow, one daughter strand is synthesized continuously, while the other daughter strand is synthesized in pieces. The pieces are joined together by the enzyme DNA ligase. Eventually, all the replication bubbles merge, yielding two identical DNA molecules.
And you'll notice we have our enzymes that come in, our RNA polymerase. Okay. So what do I need to remember? It comes in, it makes a copy. When it separates, this is a complete blueprint, and this is a complete blueprint, and each one is going to go to a new cell.
Here you can see the simultaneous formation of a leading and lagging strand. A DNA polymerase assembles a continuous leading strand, while primates, other DNA polymerases, and ligase all work together to make the lagging strand. The enzyme helicase continues to untwist the double helix, exposing more template strand DNA for replication.
You don't have to know at this level that it goes from the 5' to 15. When you are going on, then you will have to memorize that. At this level, you don't. Okay. So, parent DNA, daughter strand, when they are reforming the nucleus for two new cells, semi-conservative, right? They don't split back apart, they just stay with the new partner. Okay. Ribonucleic, now that was talking about DNA. Now let's talk about RNA. Yes, it is a nucleic acid. Its building block is a nucleotide. Ribonucleic acid nucleus is a nucleic acid, but it's different from DNA, and it even has a different job. Instead of being a double-stranded helix, it's going to be one strand. So it's a single strand of nucleotide. You will have to know what is the same and what is different about DNA and RNA. So, now, here is my RNA structure. It's a nucleotide. Look, I have my 5-carbon sugar, I have my phosphate group, and I have my nitrogenous bases over here. What do you notice that is different different? Can you find something different in my nitrogenous bases? I've got uracil instead of thymine. So I have A, G, C. So when I am doing RNA, anytime where you would have put a T, you would now put a U. You need to know that, okay? So this is my RNA language. It's a little bit different from your DNA language. RNA has that 5-carbon sugar ribose instead of your deoxyribose sugar, which is in the DNA. It does not contain thymine for a nitrogenous base. Instead, it uses uracil. There are 3 kinds of RNA based on their shape and their function. Now, we said our essay question is going to be, give me the steps of protein synthesis. These 3 RNAs all have a job in protein synthesis. You will have to know them and use them and put them into your writing. We have messenger RNA, which is little m, capital R, capital N, capital A. Again, write it my way. Okay? You have your transfer RNA, which is little t, capital R, capital N, capital A. And then I have ribosomal RNA, which is little r, capital R. That's my way. And it's not my way, it's the science way. Every class is going to be it this way, but I have a couple people in here going, I'm not going to use capitals when I write my elements, and it makes me crazy, so you all get yelled at. Okay?
So is it half a point if you change the go out to the big R?
No, but I start to switch more.
So that's
Okay, differences. DNA is a double-stranded helix, both sides of the ladder. You have a 5-carbon sugar, but it's deoxyribose sugar. It's always located in the nucleus of your cell, and its bases are ATGC. RNA, can I go back? Yes. RNA has a single strand. It has a 5-carbon sugar, but this one is ribose. It's a little bit different. It's made in the nucleus, but it goes out of the cell.